Dr. Pora Kim. Dr. Kim is an assistant professor in bioinformatics at the School of Biomedical Informatics at UT Health. She's got expertise in computational biology and precision medicine based on accurate cellular mechanisms with bioinformatics and genomic data science approaches.
Yes, so I'm bioinformatician, computational biologist. So I'm working in UT Health, School of Biomedical Informatics. Last week, our school name has been changed to D. Bradley McWilliams School of Biomedical Informatics by the generous donation by Mr. Bradley McWilliams. So I hope this can be another way to improve, enhance our schools more. Anyway, so more advanced in our school. Anyway, so yeah, today my talk title is Systematic Bioinformatics Studies for the Development of Targeted Therapeutics. So here I was invited to talk about RNA related mechanisms. So I can speak about my experience, like what kind of RNA level mechanisms can be potentially targeted, or some mechanisms can be identified using RNA-seq data analysis. So first, I can share several databases which are providing some resources as the targetable mechanisms. So this image I edited from the image by Dr. Vogel. So we have this kind of biomolecular levels units like DNA or RNA, and those modification happened in DNA or RNA level, then it will be reflected to the ORF, and then it will finally translated into proteins. So in DNA, like SNPs or SNVs or copy number variation, structural variants, or indels can be detected by whole exome or whole genome sequencing. And then the RNA level, there can be like RNA editing or alternative splicing or polyadenylation can be identified in the RNA-seq data. And the fusion gene actually made in the DNA level, but if we use genome sequencing data, it's very hard to detect, because the fusion gene means if the DNA breakage happened inside of the gene body, then we call as the gene a fusion gene. But most of the structural variants happened in any location in 3 billion genome. So usually people predict the fusion gene using RNA-seq data. And yeah, so many potential mechanism studies here I share with you were initiated from during my studying fusion genes. So specifically for this one, I studied it when I was postdoc, and then I made this project. So we know that BCL-ABL1 fusion is the target of Gleevec in leukemia, but it has, after certain finding of the specific kinase domain mutations, like which are located in very specific position, which can be related to the causing of the drug resistance. Like those mutations can be located in the gatekeeper loci, or G loop, or alpha C helix, or A loop. So this kind of located point mutations can change the drug response. So from there, I got this idea. So we systematically screened among 500 human kinases, how many and what kind of point mutations are located in this kind of kinase domain hotspots. So we found several cases like here. And this was also interested in, when I see the TMPRSS2-ERG fusion, this has happened in more than 50% of patients in prostate cancer. So I thought because the TMPRSS2 gene is androgen hormone, very specific gene, which is a tissue-specific gene. So from there, I started to think about what kind of other types of genetic features existing in cancer and the healthy population in terms of tissue specificity. So I screened this kind of diverse features. And this is, a long time ago. At the time, many people thought about those metabolism in cancer. This is also one of the key factor we need to pay attention. So in there, we gathered the metabolism genes, and then we performed multiple bioinformatical analysis, and then we found some Warburg effect related events in the breast cancer pathway. And this is similarly like, this also done several years ago. The motivation was like if there can be some mutations happened in the drug binding sites, it will affect the drug binding affinity, something like that. So we tried. And this work is about showing the exon skip database. So we mainly analyzed the TCGA pan-cancer data. Motivation was we know that recently there was a report targeting MET exon 14 skipping event. The drug was approved by FDA. So similarly, we wanted to find this kind of similar mechanisms. So the left image is showing those evidence of the MET exon 14, and the right panel shows the RB1, the tumor suppressor genes, a similar exon skipping event. And this one shows the a similar analysis, but we investigated in the Alzheimer's disease. This one is, sorry, I didn't change the title. So this is about exon skipping event in Alzheimer's disease. So we found several interesting cases. And next time we analyzed the RNA editing. I think the image changed. So we try to perform the functional annotation of the RNA editing event, like RNA editing frequency and associations, and how this RNA editing can affect the RNA structure, and how to affect the gene expression, and how to alter the protein recoding. And the alternative splicing events are also sometimes their splicing sites can be made by RNA editing events, something like that. And also we investigated those RNA editing events in the Alzheimer's disease, and recently also published one paper, RNA editing events in the Parkinson's disease. Yeah, and this was with one of the colleagues. He found a large number of correlation, of positive correlation between microRNA and genes. So he focused on this analysis in TCGA. So except this, this is our group led by Dr. Xiaobo Zhou in our center in SCBMI. He's a mathematician, but he studied very hard, and then many students are developing many interesting databases. And one of the recent one was spatial transcriptomics single cell data database, and the other one was the aging database. So from there, we analyzed and found some features relating those issues. So from now on, I want to share my experience how to target the fusion genes. So this was my topic since my PhD course. So this slide shows the concept of the fusion gene. So I can say that fusion gene is kind of a byproduct of the structural variants. So we know that in the DNA level mutation, there are many different types of variants according to the length of the size of variants. The shortest one is SMV, and the next one is indel or copy number variation, and maybe the largest one may be structural variants. CMV also can be one of the structural variants. So this is a cancer cell, and inside the nucleus there is DNA. So due to certain factors like epigenetic factor or any chemical, there can be happened like DNA double-stranded breakage at the same time in two different located genes, and then our cell tried to repair this event, but it failed. Then After that, those who failed and the rearranged chromosome will follow to the central dogma. So it will make fusion transcript, and then after translating, it can make fusion protein. So this is the example of a BCL-ABL1. So if the imatinib is binding with the pocket of ABL1, then you can you can inhibit the very active function of kinase domain. So, yeah, so here I summarize the potential mechanisms, how the fusion genes are making tumors. The first one is the widely used mechanism, signal transduction without control in the case of kinase fusion genes. So this is well studied so far. There are many drugs. And the second one is maybe target gene activation through the DNA binding domain, retained transcription factor like fusion proteins. And sometimes like MLL fusion. Actually, this MLL is wild type, have interaction with the very important cellular regulators. But if it form fusion, then it can lose this bind interaction, then it can make cancer. Or sometimes like in glioma patient, there has been reported like FGFR3 and THCC3. So this is also one of the kinase, but people identified if after formation of this fusion, at the end of the first genes, like 3' UTL, it can lose the microRNA regulation. So this microRNA is very the representative regulator for this gene. So as a result, the kinase function can be enhanced. So the other one is like if the fusion protein affect to the cellular and very important downstream effector, then this effector can affect to their target. So if we can target fusion also plus downstream effector together, then it can be better. Or sometimes one of the subunit of important cellular complex also can form fusions, and then naturally it can lose its usual function. So during my PhD course, I made some tool to predict the fusion gene from the Sanger sequencing and the 454 flex data and the Illumina sequencing data. So we made this one very early, but we wrapped up recently. Anyway, applying this approach to many NGS data, we found that these kind of novel fusions, ARC-PTPN3, we found in the H2228 cell line, which is the EMF ARC positive cell line. And the BAMP2 NRG1, we found the non-small cell lung cancer. And we also found the interesting fusion gene CTLA4, CD28 in T-cell lymphoma. So, and then since 2006, I'm involved in many fusion gene knowledge bases. So these three ChimDB 1, 2, 3 were made by my supervisor, and the fusion gene database is made by me. So this is the first database and the unique database to provide the comprehensive functional notation of all existing human fusion genes. So I am involved in these databases also. So recently we made a Google page report site. Then we can see that all over the world are using our database. Yeah, so this slide shows the overview of the fusion gene database second version. So firstly, we provide all, so per fusion gene, we provide all this annotation. So first, we provide our unique gene assessment score, and then we provide some potentially functional group. And we also show where is the most frequently used breakpoints. And the for BCD annotation, each of those we made some tools. So to provide fusion gene genomic features, we made a fusion AI model. I will introduce that later. And then to provide, to check the coding potential, we made a deep WhatF. And we made a FG viewer, which is the fusion-specific functional feature visualization tool. And then we provide the full-length fusion transcript sequence and the fusion amino acid sequence. So these are only available in our database. And these days, the UniProt team is importing our data to the UniProt. Yeah. So in our database, we have 102,000 fusion genes. And out of these, when we check the open reading frame, like 16,000 were in-frame fusion genes. This is the FG viewer. So if we put fusion gene name, then it shows the breakpoint, and then these are the known breakpoints. And then if we move these two lines, so the left first gene, right part is the second gene. So if we move these two lines, then we can infer this gray shaded error will be truncated in terms of function. So firstly, we show the what kind of TFR in there in DNA level, and what kind of microRNAs are relating each of these genes in mRNA level. And the protein level, we show the UniProt's 39 functional features, and we show the pathogenic level, including ClinVar. And this is the Fusion AI prediction method. So we focused on the exon junction breakpoints because so far almost of the fusion genes are predicted in RNA-seq data. So we made the training data based on exon junction positioned breakpoints only. And then we made negative data like this based on my experience to make those tools. And then we can see that based on the different lengths of reading for the training, we see that almost of the accuracy is similar. But we wanted to find some feature in the breakpoint area, so we'll use the longest one. So which is we are reading a total 20,000 nucleotides, which is plus or minus 5 flanking region from the 2 breakpoints. And this is the overlap with the 44 types of genomic features across BCR-ABL1's high feature importance score regions. So these are showing the top 1% feature importance score regions. Yeah, so by invitation of STAR protocol, we made this, published this protocol. And utilizing this approach, I thought Fusion AI Fusion AI is including the key features of a fusion gene breakpoint. So the usage of Fusion AI is like even we have bulk cell or single cell, anyway, we have to run the typical NGS-based prediction tools. But the key is every time we have lots of false positives. So you can think like Fusion AI can help to reduce false positive efficiently. Anyway, so here there was many reporting like micro MMEJ mechanism, which is a micro homology mediated end joining event. It can make many mutations, including chromosome rearrangements. So based on the context, I thought this Fusion AI can be helpful to identify microhomology sequences around the breakpoints. So we focused on top 50% of the feature importance score region around the fusion breakpoints. So we screened all of those, and then we found a certain amount of microhomology. And then I found, after performing several multiple bioinformatics studies, then I found some relationship between microhomology and some immune system. So recently, we also predicted fusion neoantigens from our fusion protein sequences. So when I checked the relationship between microhomology and the neoantigen, it seems there is some certain relationship. So RPS, BMP2 in breast cancer, BCR-ABL1, just kind of having a high relationship. So this one is showing how to predict the coding potential of human fusion genes, which is important because we want to find the protein level coded fusion proteins so that we can target those proteins. So we use this kind of approaches, and then we adopted the existing best approach, and then we focused on using training using more reliable data from the real ribosome data. So then our model improved a lot. And then recently, from last year, I'm working this project. I really hope this published soon. So now we have 43,000 fusion protein sequences after these kind of steps. And then among these, we choose like 1,200 like manually curated fusion protein. And these 2,300 are those fusion genes are having at least more than 2 samples recurrent fusions. So for these, we predicted the whole structure. And then out of these, we selected some reliable cases. Then for those, we performing virtual screening. So this is the PLDT score distribution of the predicted fusion structure. So somehow we see that because the currently usual prediction, protein structure prediction or reliability score, all are just focused on wild type, not fusion. So it was hard to provide more evidence, but we tried our best. So this is, we are thinking how we can find the best interacting molecule for fusion, not wild type. So we are trying many different ways, but this is first filtering scheme. So this is the EWSI1FRI fusion in Ewing sarcoma, which is a pediatric bone cancer. So this is a fusion proteins PLDT score, and this usually this position there is the functional domain, and this blue dots is we predicted the best active sites based on this predicted structure, and this is a comparison between active site PLDT score versus non-active site PLDT scores, and this is we superimposed the fusion wild type. 2 of these and the broken proteins, those 5 molecules. So we are trying, we hope to see, can find some interesting molecule. And you may know, like several years ago, there was FDA approved for the NTRK fusions kinases. So just we wondered whether our assumption is right or not. So the first one is our whole structure, fusion whole structure based predicted active sites. The second one is just the domain-based, the typical way, domain-based active site. You can see that specifically NTRK type inhibitors have a lower energy, which means high interaction in our way. And more recently, we are predicting the fusion neoantigen. So because the neoantigen is these days, many people are studying, and it can be one of the way of therapeutic of the fusion. So if we focus on this breakpoint area, I mean, anyway, so after using all this kind of proteins, wild type or fusion protein, it will be degraded. And then after degradation, then there will be some short peptides at the breakpoints. So if it is presented in the cell, then the immune system will attack those cells. So we are predicting all, and then we found like 20,000 unique breakpoint area. So we hope to make another version of AI approach. So this is a way of assessment of genes in kinase fusion network using PCA or VAE, like feature reduction. And this is one interesting study that we are doing. So for the 4 representative transcription factors, we try to map the breakage map in the protein level. So we will make for 100 proteins during summer. Yeah, thank you. This is all my talk. Yeah, here are my collaborations, my members. Thank you.